In this activity, we move from ideas and hypotheses to actual data. This is where the science really happens. Careful measurement, careful recording, and making sure our data are good enough to support real conclusions. Measuring one leaf would tell us almost nothing. Plants vary, even within the same species. By measuring multiple leaves, our replicates, we get a much better picture of what's typical and how much natural variation there is. Replication is what turns a single measurement into real data. In this experiment, we're collecting 10 leaves. When growing plants, you would typically do 4 to 10 replicates per treatment group. You would do closer to 10 if you're growing from seeds, because not all seeds germinate compared to if you're buying seedlings. You would place 1 seed per cell unless your experiment is to test the effect of crowding. If you are doing a germination test, such as seeing the percentage of seeds that germinate based on a treatment, I recommend doing 4 replicates of 20 or 50 seeds. One of the most important rules in experimental science is this: you only change one factor at a time. Everything else has to stay the same. That way, if you see a difference in your results, you can confidently say it's due to the factor you changed and not something else you accidentally let vary. Let's use the fertilizer example again. If you want to know the optimum amount of fertilizer for a specific species, then everything else has to stay the same. Same species, same light, same water, same soil, same pot size. The only thing you change is the amount of fertilizer. That's how you isolate cause and effect. If you change more than one thing at once, say fertilizer and light, then you won't know which one actually caused the change in growth. Good experimental design is all about controlling variables so your conclusions are actually meaningful and defensible. Look at the image on the slide. It has plant A that had fertilizer added and plant B that did not. Based on what you learned in the previous slide, what is one error that was made? If you said that it doesn't have replicates, you would be correct. Also, in the experiments that you all create in this course, you will have 3 different treatments. You would have the control, which is a standardized, unchanged group that is used as a benchmark to compare with the experimental groups. In the case of the fertilizer experiment, it would be a plant with no fertilizer. In the leaf area experiment, we don't have a control group since we are looking for differences between species. This is where controlling variables shows up in a very practical way. We're not changing fertilizer or light here, we're controlling how we sample. You're going to collect 10 mature, undamaged leaves that are simple, not compound, from 3 different plant species. You don't need to know the name of the plant. I don't want you collecting leaflets, but leaves. If one species gets young leaves and another gets old leaves, any difference we might see might just be due to age, not species. The young leaves are typically lighter in color and smaller, located toward the end of the branch. You will want leaves more in the middle of the plant. You will need to pick the leaf with the petiole intact. Although we won't be measuring the petiole, tearing it off may remove too much of the leaf for accurate measurements. Consistent sampling keeps the comparison fair. For this lab, you'll also need to document your samples with photos. You must take 1 photo of each set of leaves. That means 1 photo for species A, 1 for species B, and 1 for species C. Each photo should clearly show all 10 leaves for that species laid out so they're visible and not stacked on top of each other. The goal here is simple. I should be able to look at your photo and see exactly what you measured. Make sure each photo is labeled, either with a piece of paper in the picture that says species A, B, or C, or with the actual species name if you know it. This is your proof of sampling and your record of the material you used. Think of these photos like part of your lab notebook. They help document that you collected real samples, that you used the correct number of leaves, and that your measurements are based on actual data. You'll upload these photos along with your lab file when you submit the assignment. If your photos are unclear, missing leaves, or not labeled, it becomes possible to verify your data, so take a minute to do this carefully. It'll save you a lot of trouble later. Consistency matters here. Measure the same way every time. Small differences in technique can turn into big differences in your results. You should measure the length in centimeters. I follow the mid-vein. Make sure you do not include the petiole. For the width, measure at the widest point. Use Excel to organize your data. You can copy over the example if you prefer, but keep in mind that when you do your own experiment, that you will need to create your own tables. Each table for each species will have a column for the leaf number, width, length, and leaf area. You are not required to have the species name on your tables like I do. In the table, you'll include the width and length for each of the 10 leaves for the 3 species. Now that you have your width and your length placed into the Excel spreadsheet, we're going to calculate the leaf area. So instead of calculating it by hand, which area is length times width, we're going to have Excel do it for us. So we can do that by performing a function. To perform a function, you start with the equal sign here. And then you are going to you're going to calculate length times width. So we have width here, we have length here. So width is B3. And then the time sign it looks like a star. And then we have C3. And then you press enter. And that has now given us our leaf area. So instead of doing that for each of these, we're just going to go back to this one, and we're going to drag the corner here. And there we go. And we have that all filled in now. Makes a shortcut for us. So I've already calculated for the other 2 tables, but if I click on here, you can see it says equals G3 times H3. This one here says equals L3 times M3, and I dragged a corner for each of those to calculate it. The next thing we're going to do is calculate the mean. The mean is the average leaf area. So when you add all these up, all the leaf areas up, and divide by 10, you get the average. It's good to look at an average. We're going to be plotting the average for the graph. Instead of having each of the data points, it's better to have an average. In order to calculate the average, you click on this square, you're going to press equals, and then start typing the word average in. So here we have the average, and then this time it's going to be D3 colon D12, and then in parentheses enter, and there's the average. The same was done with the other ones. You can see here it says I3 colon I12, and this one is N3 colon N12. Standard deviation tells us about consistency. So 2 species could have the same average leaf area, but one might be very uniform while the other is all over the place. So standard deviation tells us how spread out those numbers are. And that difference can matter scientifically. It's actually something you can explain about if there's a large standard deviation. In order to calculate the standard deviation, we'll click in there, start with the equal sign, and then we put ST, and then it'll pop up a whole bunch of them. We want the one that says stdev.s, and then this one is going to be D3, D12, so that same range as average, and click enter. And this was done the same for the second one. You could see there I3, I12, and this one is N3, N12. So what does this mean? You notice that species A has 4.34. There's almost to 1.99 for species B, and 1.48 for species C. But we notice the average is the same for both B and C. So they're similar, but does this mean species A had a much larger standard deviation? In this case, not really. If you calculate how much the standard deviation is compared to the average, it will give you a better idea. So for species A, the standard deviation is about 18.5%. So what I did is I took this number, the 4.34, divided by 23.35, and I multiplied it by 100 to give me a percentage. That was 18.5%. If I did the same for species B, I get 18.3%, and species C is 13.6%. So, okay, they're kind of similar. So what does that mean? Well, if your standard deviation percentage is over 30 or even close to 50, then that would tell you that you have some inconsistent results. This could happen if you had some young leaves and some old leaves in this case. But when it comes to your experiment, that could mean different things as well. So that's why standard deviation is important. It tells you how large a spread you have in your values. The next step is going to be building a summary table. Since raw data is messy, we're going to turn those 30 measurements into something that you can compare visually and statistically. So this summary table is going to help us with our graph. So I'm just going to put the averages in here. And the standard deviations. There we go. And then we're going to create a graph for this. So the graph will show us different patterns instead of plotting all 30 raw values, because that will be cluttered and it's hard to interpret. So the way we're going to do that is we're going to highlight just this part here, the species and the average leaf area, and then we're going to click on Insert and then Charts here, and we're going to click on this top one. That's 2D. So there's that one or that one. They look about the similar. I like the wider one here. So if I click on that, oops, I'm going to move this down here. So we may want to add some things. So I actually have another one that I kind of created here. So I changed the title to average leaf area for different species. I could say for the names or species A, B, and C. Let's move back. So I could change this to that one here. We also want some titles here. The way you're going to do that is click on the plus. So axis titles. We can also have some data labels, and we're also going to be doing some error bars. So first, the axis title, let me see what I changed this one. I put leaf area in centimeters squared. So I'm going to copy that over to this one. There we go. For this one, I'd probably just take it out because it doesn't really lend much. You can't really see the number here. It's not required to have that. So I'm just going to change it by right-clicking format. If I change the fill, we'll see if this works. I changed it to like a lighter color so you can see it. So you can see those data labels a little easier. And then we have the error bar. Right now the error bar is just kind of a random number here. I'm not real sure what it has it in there. It says standard error. Well, we actually have the standard deviation is the error. So how do we change it to these numbers here? We're going to go to custom and then specific values. And we have a positive and error value. So in order to get that, we have to, let's see if this will work. There we go. So I highlight the standard deviation in that little table, and then you do the same for the negative value and click OK. And now we see that this one's larger than these two. These two are, this one's a little bigger and this one's a little smaller because that's what standard deviation was. So that you'll do that in your own experiments as well. Before you answer these reflection questions, I want you to think about why we designed the experiment the way we did, not just what steps you followed. For the first question about collecting leaves of similar size and position, think about fair comparisons. Ask yourself What would happen if one species was measured using young leaves and the other using old leaves? Or if some leaves came from the top of the plant and others from the bottom? Would any differences you see really be due to species? Or could they just be due to how you sampled? This question is really about controlling variables, so your results are actually interpretable. For the second question, about standard deviation, remember that the mean tells you what's typical, but it doesn't tell you how spread out the values are. Think about the difference between a group of leaves that are almost the same size versus a group where some are very small and some are very large. Standard deviation is about variability and consistency within a species, not about which species is bigger. For the third question, about why we graph the mean instead of every single measurement, think about the purpose of a graph. A graph is meant to show patterns clearly and make comparisons easy. Ask yourself what the graph would look like if you plotted all 30 individual values and whether that would make the pattern easier or harder to see. The mean helps summarize the data so the main trends stand out, while the variability is still handled by statistics like standard deviation and ANOVA. The goal of these questions is not to repeat steps from the procedure, but to show that you understand why scientists measure, sample, summarize, and analyze data the way they do. The main takeaway from this activity is that good science starts with good data. Careful and consistent measurement is what makes your data reliable in the first place. If measurements aren't done the same way each time, then any conclusions you draw later become questionable, no matter how good your statistics are. Replication is also critical. Measuring 10 leaves per species isn't busy work. It's what allows us to see what's typical and what's just natural variation. Replicates let us calculate meaningful averages and understand how variable the data are within each species. Controlling how we sample is another form of controlling variables. By choosing similar, mature, undamaged leaves and measuring them the same way, we make sure that any differences we see are more likely due to species and not due to how the leaves were chosen. Finally, using the mean and standard deviation and then summarizing the data in tables and graphs helps us see patterns clearly and prepares us for the statistical tests we'll use next to decide whether those patterns are actually meaningful.